In this figure, we're going to look at membrane structure, and specifically, we're going to focus on the phospholipid bilayer. And really the only difference is that the membrane is a more complete structure with embedded proteins and cholesterol and other molecules. We're just going to focus on that backbone, the phospholipid bilayer. And that's comprised of many, many individual phospholipids. We're going to take one of them over here and blow it up so we can look at its component parts. In the middle here, we have this central glycerol backbone. And two of the carbons of that glycerol molecule, we have hydrophobic fatty acid tails attached. This is the water-fearing end of the molecule. Water-fearing, hydrophobic, like a phobia is a fear, right? And so it's fearing water. And what that means is that thermodynamically, these hydrophobic tails will all become associated with one another. And so by excluding water, this guy will associate with the hydrophobic component of this guy, and so on and so forth. And we generate this spontaneously forming lipid bilayer due to this exclusion of water from this hydrophobic core. Now, if we look at the other end of the molecule, we find a nitrogen group and we find a phosphate group which has a charge on it. Both of those things are really important to the fact that this end of the molecule is water-liking, right? It's hydrophilic. This means that it readily associates with the polar nature of water. And overall, our molecule now has a water-liking end and a water-fearing end. And together, those two create a molecule that's known as amphipathic. So that characteristic is absolutely essential to this formation of phospholipid bilayers because they spontaneously form with the hydrophobic core in the center away from the water on either side, and then the phosphate heads and the nitrogen groups sort of facing the aqueous solutions. Now, why is this important? Well, it's important because it helps provide a semipermeable membrane. What we mean by that is that certain substances will pass readily through this membrane while others will not. The ones that pass readily through are very small and tend to be hydrophobic. So things like oxygen or carbon dioxide or other small or some small lipids will readily pass through this membrane. They tend to be able to squeeze in between the phosphate heads and then readily associate with the hydrophobic tails, and then they can just sort of diffuse their way out on the other side. And you might think that they'd be repelled by the polar heads here, but there's enough space in between that small lipophilic substances, same thing as hydrophobic, would be able to pass readily through the membrane. This is important because it allows oxygen and carbon dioxide to readily move in and out of cells and in and out of organelles. Now, larger fatty substances are actually repelled by this polar heads of the phospholipids, and they actually require transport proteins to get across the membrane. That puts them in a category with the polar substances, small or large, things like glucose or amino acids. We'll put some of these substances out here. Those would be on one side, and because they're polar, readily associate with the phosphate heads, but can't associate with the hydrophobic core. And so, they'll come down here, associate, but then just be repelled. This means that we can maintain gradients of these substances. If a cell takes up glucose, that glucose would not then diffuse readily right back out of the cell. How does the cell take up glucose then? Well, we have to have specialized transport proteins for our polar substances, things like nutrient transporters or ion transporters. Charged entities fall into the same category. They'll associate with this polar head, but they cannot associate with the hydrophobic core. Right? And this is all really important because the phospholipid bilayer allows us to maintain gradients. In the absence of these transport proteins that we're talking about, we can maintain ion gradients, we can maintain nutrient gradients, gradients of waste molecules that are not readily allowed to pass passively through this membrane. Once again, we do have transport proteins that we'll talk about later that allow many of these substances to move across the membrane under controlled situations.